So hello everyone. Thank you very much, Laurent, for accepting this invitation from ALPACA and PANGAIA groups that are both working on pangenomics. And so Laurent Jacob is a CNRS researcher working in Lyon at LBBE. And he works on GWAS, on phylogeny, and what did I note? On prediction of phenotypes with machine learning, if I am correct. Mm-hmm. And today you are going to talk about one exploitation of the Bruijn graphs that we are not used to. So use the components of the Bruijn graphs as variants in the bacterial world. So thanks again for accepting this invitation. And the floor is yours now.
Thanks a lot, Pierre, for the very kind invitation, and thanks everyone for attending. I'm very happy to have the opportunity to discuss that with you. And so, yes, the context, like Pierre recalled, is the relationship between genotype and phenotype, and more precisely, GWAS, which are genome-wide association studies, and that aim in general at finding correlations between genetic variants and trait, a phenotype. So it's a very broad scope that includes genetic risk, plant breeding, and the general procedure for that is to build a table for each variant. We have a set of individuals. And for each individual, we know if the individual has or doesn't have a genetic variant. So we build this table for each variant, and then we test the association.
Laurent?
Oui.
Sorry, I'm not sure we are seeing your slides.
You don't see the slides?
I'm sure we are not seeing your slides, actually.
You don't see at all, or you don't see them?
No seeing anymore.
Okay.
So we saw it a few minutes ago.
For some reason, it stopped. I don't know why. Do you see it
now? That works. Yes,
okay.
It works now.
Okay, let me know if it stops again. Yeah. So yes, you build this table. You have a set of individuals for which you know who has and who doesn't have the variant. And for each person, you also know the trait of interest. Is it a control or is it a case? And once you have this table, you just test the association between the presence of the variant and the trait with your favorite method, which can be a linear model or a Fisher test, for example. And so what you see in all this explanation, I put it in red here, is that all of this depends of the definition of a variant. And very often it's not questioned at all, because what's a genetic variant is something obvious. And this is because historically GWAS were mostly done for humans. And in humans and other similar cases, the standard approach is to use SNPs, short nucleotide polymorphisms, or short insertion deletions to define variants. And this is because, as you probably know, all human genomes are almost identical up to this small variation. Like sometimes one letter will change, or there will be a short interval. So in this case, using this definition of variant is a very, very good option, because it will capture, it's simple to define. You just have to align all your individual genomes to reference. It will capture all the relevant genetic variation, and then you can put it in your GWAS pipeline. But there will be other situations where this is not true anymore. Even if you just remain within humans, if you don't have healthy human cells, but you have tumor cells, for example, then there will be, for example, translocations, and you won't capture translocations just by looking at SNPs. So you will miss part of the important genetic variation. And where I'm going with that is that it's even worse when you look at microbes, because SNPs or indels are only well defined over what's often called the core genome in this context, which is the set of genes which are shared by all the individuals in the panel, all the samples in the panel. And the problem is that in lots of cases of interest, like when you study drug resistances, it's also important to look at the accessory genome, which is sets of genes which only belong to some of the genomes in your panel. And for lots of drugs, the non-resistant genetic determinants are in this accessory genome. So if you just look at SNPs, you don't even look at the accessory genome. You can only define it on the core genome. So what people have been doing is they have added this information to the list. They say, okay, we'll look maybe at SNPs, and we'll look also at the presence or absence of lists of genes in the accessory genome. So you can do that, but then what about non-coding regions, like parts that are not genes, like promoters, for example, enhancers? Or what about SNPs in accessory genes? And what you start seeing here is that you can always answer, okay, I will just add that to the list, right? I know this SNP in this gene can be important, so I'll put that in the list. You can do that, but then you're always limited by the variant list. And so what's important is that you will need to start with a list of candidates that cover all relevant variants. If you don't have them, if you don't have them in the list, you won't even test them, so there is no way you're going to find them. So because of that, an alternative approach has been to use k-mers instead of to define the variants instead of SNPs or genes. So you're probably all familiar with k-mers. K-mers are words of size k in the ATCG alphabet if we look at genomes. And so instead of saying, I'm testing the presence of this SNP in the genome, is it related to resistance? We are going to say, I'm testing the presence of AA, T, C, C, etc. And we're going to do that for all possible words of size k in the genome for some value of k. So great advantages of that are that it's very expressive in the sense that it will capture SNPs, presence of genes, translocations. All of this will somehow trigger the presence of a particular k-mer. Having this k-mer will be related to having this SNP or this gene in the genome. And it's doing all that while remaining what we may call agnostic, in the sense that you don't need to have a predefined list of SNPs or genes. You just say, I'm looking at all the words in the genome. So even if you have an alien strain that arrives, you just have to sequence it and you can define it. You don't need to have any annotation. You don't need to have a reference, and you don't need to align anything to do that. So the flip side of that is that you lose a lot of interpretability. It's very tedious to interpret the results. That's how we started working on that. Is people told us, yes, that works in the sense that you can run it, and then you get a list of thousands of k-mers, and the hard work starts at this point. So some intuition on why it's hard to interpret, and you get part of it is that you get a lot of redundancy, because several k-mers will often represent the same determinant. So imagine that what's important for drug resistance is to have a SNP in the genome. Then in this case, if you look at k-mers, all the k-mers that overlap with the presence of the C or A in this case, all of these will be associated with the phenotype. And if you do the test, and then you will have to work your way back from this big list of 8 k-mers to see, oh, okay, this was a SNP somewhere in a genome. Similarly, in some cases, if what's important is to have a gene, a resistance gene, for example, but this gene doesn't have the exact same sequence in all your samples, then it may be hard to have, you won't have a single long k-mer that will represent the gene. So here it's a very short gene, as you can see, but there is some variation. So you will have the choice between having short k-mers like ATC or CAAG that will be conserved across all the versions. So then again, you will have to work your way back to a full gene. Or there will be different versions. If you take something larger, you'll see that there won't be a single version of the gene. So it makes the interpretation difficult. There's huge list of k-mers, and also I will discuss that it can dilute the effect when you lose some power when you test, because instead of having a single test representing the presence of the gene, for example, you have different samples containing different versions will be considered as different variants. So a general approach that we've been taking to deal with this problem has been to use the Bruijn graph to structure the genomic k-mers. So probably lots of you know very well the Bruijn graph, but I will recall what it is. It's a graph that connects overlapping k-mers in the sense that the nodes of the graph are the k-mers here of size 4, and you draw an edge if the suffix of one, like here TCG, is the prefix of the next one, here TCG or TCG. And it's something that's it's an old and very widely used tool, in particular for genome assembly and variant calling methods. And here it's something we've used. We've noticed that. So if you're familiar with the Bruijn graph, you see that here you start from TTCG with the same example before with the single SNP. This SNP will create a fork because you have two different k-mers that are connected to TTCG. And if you keep adding k-mers and building the graph, you see that all the k-mers that contain the C will be added on a single path, and all those that contain the A will be linked on a distinct path, and they will be connected on k-mers that don't contain the mutation anymore. And once you have done that, you can do what people call compaction. That is, you will notice that all those that contain the C are on a single pass, and so instead of testing separately all these k-mers, you can say having any of those k-mers in my genome will be equivalent to having any of the others. I know that if I have one, I have all the others. So instead of testing these short k-mers, I can do a single test for a longer one, which is called a unitig in this case. So this will remove part of the redundancy I was talking about. Pathways will be compacted into a single longer sequence. It should be easier to interpret, but it will do more than that because we see that it's likely that it's not always the case, but k-mers that are connected in the graph are likely to be in neighboring regions of the genome. So it will represent some genomic context, and it will help us get some interpretation from the k-mers. So this is and this is not exactly what this presentation is about, but this was a previous work. The first step maybe for that is a work that was done by Magali Jaillard and Leandro Ishii during their PhDs. It was a tool called GWAS, and in this tool, we only used the Bruijn graph for interpretation of the k-mers. We tested the k-mers and we tried to make some sense out of it with the Bruijn graph. This is the idea. If you build the single the Bruijn graph on all the k-mers that are present in all the strains, the strains of the genomes of the strains in your panel, you get a big ball like this. So it's not like the small cartoon I had before. It's not, you cannot really inspect it k-mer by k-mer. It's way too big. So what we do is that, sorry, remember that the variants, what we are going to test are the unitigs in this global Bruijn graph, so presence or absence of a short sequence in the genome. And we're going to test all of them. Typically in the data sets we had, it was about 2 millions. We had 2 millions of unitigs. So you're going to do 2 millions of tests and identify those whose presence is significantly associated with the phenotype. So it's a few dots in this big ball. And to help interpret the results, we are going to add the neighborhoods of these identified unitigs. So we are going not only to look at these short sequences, but also those that are connected in the Bruijn graph because they are overlapping. And you see that if we do that, some of these neighborhoods will overlap. So maybe these 3 here unitigs, they were not directly overlapping, but their neighborhoods, they are in the same extended neighborhoods. So once we can see that once we extend to the neighborhoods, and we are going to return, the tool is going to return connected components obtained by extending to neighborhoods like that, which we are going to annotate and color to get some interpretation. And so this is the typical type of result that we get. And you see that in this case, it will capture genetic determinants that can have, so here in this case, it will be a sequence of a gene, for example, but that exists, like I was explaining at the beginning, in several versions. It's a gene, but not all strains in my panel have the exact same sequence for this gene, which makes that it was not compacted when we built the Bruijn graph into a single very long sequence. You see there were lots of small bubbles here who all correspond to some variation, either SNPs that are neutral or maybe sequencing errors. Okay, so this is something that we can see with some training just from the Bruijn graph that we obtain. And sorry, I didn't mention the color here is related to just the number of resistance strains that contain the unitig. Okay, so here, remember that each node is a unitig. It's a short sequence, or not so short after compaction. And it's very rare if this sequence is contained in a lot of resistance strains. So here, this example is the top component that's written when we do a GWAS for Staphylococcus aureus resistance for methicillin. And we will cover a very well-known resistance gene. So this is the sequence of the gene here, and here it's a cassette that includes the gene. So I won't detail more than that this tool, DBGWAS, but we spent some time to propose a tool that was as easy to use as possible. You don't need a reference genome or predefined list of variants. You just give your FASTA files and phenotypes, and you get these kind of nice subgraphs. So sometimes people say well, these subgraphs are not that interpretable, which I understand. But just remember that it may still look cryptic, but the alternative at the time was to have a big list of thousands of unstructured k-mers. So for me, it's still a benefit. But if we circle back to my original argument or promise, I said we're going to use the Bruijn graph to structure the genomic k-mers. How did we really do that? So we did it in some sense, because the graphs that we obtain, they provide a higher level interpretation, right? I said with some training, you will see that these kind of almost linear subgraphs are genes or plasmids. But the statistical test that we're doing is still done at the unitig level, where the interpretation at the graph level comes after, which is maybe not very satisfying from a statistical point of view, because we do a test at some scale and then we give an interpretation at a different scale. It's okay, but it's not completely satisfying. And also, like I said, that there may be some power leak in some cases, because if what's important is to have this gene in the genome, then maybe I will never test something which is present in all the resistance, on all the strains that contain this gene. I will do one test for some of them that contain one version, another test for some other subset of them that contain another version, and it will be less powerful. And so this is what we tried to address with Hector Roux de Bézieux during his PhD, to actually do the test at the right level. And so here I'll get back to the same example, motivating example. I'm having an even shorter gene than before, just for the sake of the explanation. It's a 4-letter gene, and you have several versions of it. Okay, this is your causal gene. If you do the Bruijn graph on it with k-mers of size 3, this is what you obtain here. And you see that, so you have 4 possible unitigs. There is no compaction here at all. And you can do a test the association between the presence of each of these 4 unitigs and the resistance phenotype. And you see that here in my panel, I have 4 resistance samples. Each of them contains one of these versions. And then I have also sensitive samples that don't contain any of, that don't contain the resistance genes, so they don't contain any of these unitigs. And you see that if I test these unitigs, each of these 4 will only account for half of the resistance strains. TGA will be present in this strain and this strain, but not in the 2 others, etc., etc. So it may work because there will still be some association. And so in the best case, what will happen is that they will be detected, they will be found associated, and then if I look at dbGWAS, I will the output of dbGWAS, it will use this graph and it will show, okay, I have these 4 unitigs that are connected and that are all significant. So maybe they correspond to a single polymorphic gene, right? This is what I showed before. But you also see that ideally what we would like to test in this case is A or T, then GA, then A or T. And we have more power to test that because it's, well, in this idealized case, the presence of these new variant is the exact same presence as the resistance phenotype. And so to build it, technically, I'm just taking a logical OR over the 4 other unitigs, the 4 unitigs in this particular subgraph. So it becomes a bit abstract to say that this subgraph is a variant. When I say this subgraph, I have this subgraph variant, I mean I have any of the versions of the long sequence because I have any of the k-mers in the corresponding subgraph of the Bruijn graph, okay? So this was for one single idealized example. If I want to do that in an agnostic way, like we were doing before, otherwise there's no point. I would have to do that for all possible connected subgraphs in the Bruijn graph. And remember that in my not so big examples, I already had 2 million of nodes. And the number of connected subgraphs could be exponential in the general case in the number of nodes. So if you know anything about multiple testing, you see it will be in trouble because there will be way too many tests and there will be no power left to detect anything. And even not talking about tests or power, just enumerating all these subgraphs is probably not a good idea. So here I'm just summarizing the idea that we are going to use to solve these problems. The trick is that some of these subgraphs we'll be able to discard them without even testing them using a concept called testability that I will detail after. The very intuitive idea is that if you have a variant in general, not even forget the subgraphs, if you have a SNP that's present in all the samples in the panel, then you know there's no way it's going to be associated with anything on this panel, right? That cannot be correlated with anything because everybody has it. So you know you don't even have to test it. And intuitively, this will remain true even if it's not present for everyone. And the concept of testability will generalize that and will give a well-defined way of using this. So this will solve maybe the statistical issue. And following that, some of the subgraphs, because of the way we build them, may not even need to be enumerated. Because remember that the way we will build the variant from the subgraphs is by taking a logical OR over the colors, the presence pattern of the unitigs in the subgraphs. So if I start from a unitig or another pattern, which is this, for example, if this is not testable already because it's already present in too many strains, and I do a logical OR with anything, I just add a neighboring unitig, then it will be even larger. So probably I know I don't even need to test it anymore. I can stop exploring from there. Okay. So this will partly solve our 2 issues because the non-testable subgraphs won't count towards correction. And we'll use this pruning strategy to not explore all the possible subgraphs. So the idea of testability was developed by a researcher called Taron. And I will just here give the summary of the idea. The informal idea, like I started to give, was that you may have heard. I will recall it after, but what's very often called for for multiple testing correction is called the Bonferroni procedure. It controls the FWER at some level by thresholding the p-values. If you want to control at level alpha, you will threshold the p-values at alpha divided by the number of tests that you are doing. And like I said, when we correct for test multiplicity, here some hypotheses won't count, so we won't have to divide that much. It won't be that small in the end, the threshold. And so here I already gave the intuition that adding nodes to subgraph variants will only increase the frequency. So at some point, we won't have to test anymore. We will be able to prune. So to explain how the Taron procedure work, I will give a short reminder of how Bonferroni procedure works already. Assume that we are testing M null hypothesis. So for example, M variants could be SNPs. It was actually developed for SNPs in the first place. In our case, it will be M subgraphs. And so we have null hypotheses, which are this variant is not associated with the phenotype. We want to reject those, we want to test them. And so we are going to use some test statistics to compute p-values for each of these tests and reject the null hypothesis, each of these null hypotheses, if the p-value is lower than some threshold. And by definition, if the test is correctly calibrated, you need to remember that the probability under the null hypothesis that the p-value is lower than the threshold is itself lower than the threshold. Right? So the family-wise error rate that's correct, that's controlled by the Bonferroni procedure is defined like this. It's the probability over the conjunction of null hypothesis that any, so we assume that all the hypotheses that we're testing are null, and it's the probability that we are still rejecting at least one of them. Okay, so we're making at least one false rejection under the hypothesis that all of them were null. And there is no closed form for this probability in general. And this is why Bonferroni proposed this procedure, which is to use a very crude bound, but very general too, which is called the union bound. And it's to say, here I'm testing the probability of a disjunction or over a set of events. And we know that this is smaller than the sum of the probabilities, right? It's equal if the events are independent, but in the general case, it's smaller. And like I said, the probability of false rejection for each of these hypotheses, if the tests are well calibrated, is itself smaller than the threshold. So it means that the FWER at the threshold alpha divided by M is smaller than M times alpha divided by M, the threshold, which is alpha itself. So it means that this is why if we use alpha divided by the number of hypotheses, we control the family-wise error rate at level alpha. So that's why we use this threshold. Okay, and what we're going to use to improve this bound is the fact that discrete tests admit what's called a minimal p-values. And I'll start with a small example. Let's say we have bacteria, a certain number, 13 of them resist to a drug, and 22 don't resist to the drug. And among all these bacteria, 8 of them have a SNP in a particular gene. And this is what we want to test, the association between this presence of this SNP and the resistance. So we build this contingency table here. And we do a Fisher test. That's one possibility. We get a p-value of 3%. And then we take a decision. For example, if we test, if we are testing 1,000 SNPs like this one, and we want to control the family-wise error rate at level 5%, then we don't reject H0 because we would need to have the p-value lower than 0.05 divided by the number of tests, which is much smaller than that. Okay, so we don't reject in this case. And the question is, did we actually need to do this test to know that we are not going to reject? So to answer that, you need to see that given the margins of the table, the whole table can be deduced from one of its entries. Okay, if here I know the margins, which I was giving at the beginning, if I fix one of these 4 values here, I'm fixing the 3 others. Okay, by just deduction from the margins. So here, for this value A, I'm using the number of sensitive strains which have the SNP. So this value here. In this case, I have 2 strains which are sensitive and have the SNP. And if I fix this, I'm defining the entire contingency table. And this is the log 10 of the p-value for each of the possible values of the possible numbers of sensitive strains with the SNP. Okay, there is a finite number of possibilities given the margins. You see that's all the p-values that I can obtain. In the original table, I had this p-value, and you see that it cannot get lower than this finite value. Okay, this non-zero value. So going back to the questions, did we have to do the test? We could say, given these margins, what could have been the lowest p-value? There is an answer for that. In this case, it's using 0 for this entry. It leads to a particular p-value, minimal p-value that we call p star. And we see that we still don't reject at our level even if we have this minimal p-value. And in this framework, we say that the hypothesis is non-testable at this level of 0.05 divided by 1000. And so here I just wrote all the possible values, but in many cases, including the Fisher test, we can compute this minimal p-value in closed form. We don't have to enumerate all the combinations. So if we go back to the Bonferroni procedure, here I'm just writing explicitly the union bound. I said that we're using the fact that the FWARE is smaller than the sum of the probabilities of false rejection. Okay, so that is the probability under H0 that the p-value is smaller than the threshold. Okay, and we're using the fact that this p-value, if the test is well calibrated, is smaller than the threshold itself. So we have m times alpha divided by m. This is smaller than alpha. Okay. In our new case here, we see that for some of the tested hypotheses, the smallest p-value, the minimal p-value, p star here, will be larger than the threshold, alpha over m. And this is something deterministic. It does not depend on the data. It only depends on the margin. So because of that, from a probabilistic point of view, the p-value by definition is necessarily larger than the minimal p-value. So these hypotheses can never be rejected. And so in particular, the probability of false rejection is exactly 0. It's not just smaller or equal to alpha over m. So the consequence is that if we only have Q hypotheses left that are testable, then the FWARE is not only smaller or equal to alpha, it's smaller or equal to alpha times this number of hypotheses divided by m. So you see this is 1 if Q equal m, if they're all testable, but otherwise if Q is smaller than m, this is smaller than 1, okay? So this is suggesting that we are controlling at a better level than what we wanted to. And it's also suggesting that we could actually use a larger threshold. We could reject more and still control at the level alpha in the end. We don't have to use this very stringent Bonferroni threshold. The problem is if you think about it, there is something circular in the choice of the best threshold. It's not trivial because here we say that we can lower the threshold because Q, the number of testable hypotheses, is small, but your testable are not at a particular threshold. So if you increase the threshold, then there will also be fewer hypotheses that are not testable. The Q will actually be larger. So you cannot determine it. There is no trivial way to say, okay, this is Q, this is the number of non-testable hypotheses, because it will depend on the threshold. Okay, if you increase the threshold, then you will also change Q, and so you may not control at the level alpha anymore. So there have been methods proposed to deal with that. The idea is that you define m of k to be the number of testable hypotheses at some level, at the level alpha divided by k. And you want to find the smallest possible k such that the number of testable hypotheses is smaller than k, because in this case, you see that so Q here will be your number of testable hypotheses, m over k. You will divide by k, and so you will control, you will be smaller or equal to alpha by doing that. So here again, the naive approach will require you to compute all the minimal p-values for all your tests. And then you start at k equal 1, and you increase until you reach mk smaller or equal to k. So, sorry, not until, but as long as mk is smaller or equal to k. But okay, it works if you have thousands or a million of SNP. But in our case, we don't want to have to enumerate all the hypotheses and let alone compute the minimal p-values. We don't want to have to do that. So can we do better? In some situations, including ours, the minimal p-value will have a monotonicity property in the sense that for some pairs of hypotheses, we will be able to say this hypothesis is smaller than this second one. And if this is the case, then the minimal p-value of the second one is larger, necessarily larger than the first one. And that's, if you keep in mind the example I had, that's the inclusion for us, right? If you add unitigs to the current subgraph, then the frequency pattern, the presence pattern, will necessarily be larger. And so for some statistics, then the minimal p-value will necessarily be larger too. In our example, the SNP frequency, for example, p star after some threshold will only increase if you increase the frequency. That will also work for our subgraphs. So here's the idea that we used and that was proposed before by other papers for a faster approach. The algorithm is to initialize k to be 1, and you have an active set of testable hypotheses. And then you start enumerating your hypotheses that you want to test, and you only add the testable ones, okay? The ones for which the minimal p-value is smaller than alpha divided by your current value of k. If this set has more than k elements at some point, then you increase k, which again will change the threshold. You increase it to the cardinal of the active set, and then you may have to remove some of the hypotheses which are not testable anymore, which are not testable anymore at this level, and then you keep enumerating. So that's the general idea. And in addition to that, if you use the big interest of that is that you can stop exploring when you find a hypothesis that's not testable. You can stop exploring all the ones that come after that are larger in the sense of that their p-value, minimal p-value is necessarily larger. And so the question will be, how do we do this exploration in our case? And that's going to be the second. There were two main difficulties in our problem. One is solved by, like I said, this testability issue. Second one is that if we want to use this testability ID efficiently, we need to explore efficiently the set of hypotheses. So how do we do it? We're going to use an ID framework, which if you're into algorithmics, you probably know much better than me already. It's called a reverse search. It's a general recipe to enumerate elements in a set. The idea is that you define a reduction operation from your elements that will transform any element into a simpler one. Simpler can be anything. It's just that in the end you need to have an empty element. And by doing that, you define a tree over the set, because from any element you will get a smaller one, from this one a smaller one, etc. And in the end, the succession of reductions will lead to the simplest one, which is the root. I will give an example right after. So this is building a tree. This is defining a tree over your set of elements. And if you're able to invert the reduction operation, that is from any element, say, which are the elements that give this one by reduction, then you can explore the tree from the roots. And following this recipe, all the subgraphs that are enumerated from any given subgraph will be larger, and it will make it suited to pruning strategy because of the monotonicity property that I was discussing. So this is an example, a well-known example of how you can use this idea of reduction to enumerate connected subgraphs. And so you see that the reduction is to remove, so you use a numbering on the nodes, arbitrary numbering. And the reduction is to remove the nodes with the largest index, whose removal does not disconnect the graph. That is, here, for example, you have 1, 2, 3, 4. You remove 4 because 4 is the largest. But here you have 1, 4, 2. You don't remove 4 because otherwise you would disconnect. You'd have 2 bits left. But you remove 2, which is the largest you can remove without disconnecting the graph. And it gives you 1, 4. Okay. And so you see that this is defining, in this small example, we can picture it. This is defining a tree over the possible subgraphs, connected subgraphs, sorry. And so if we want to explore, we will start from the root, and we'll need to invert the reduction. That is saying, what's the set of subgraphs which are giving the empty sets by reduction. And the inversion is to add the node, which will become the highest index node among those whose removal retains a connected graph. That's a bit complex because of we wanted to retain connectivity by the reduction. We need to make sure that we take that into account when we invert. So for example, when we start from 1, 3, similarly to what I was explaining before, we don't just add 4, but we will also add 2. Right? Adding 4 will give a child, because 1, 3, 4 indeed gives 1, 3. But 1, 3, 2 also gives 1, 3, because here from 1, 3, 2 we don't remove 3, otherwise we would disconnect the graph. Okay. So you see that the inversion of the reduction can be a bit involved. In this case, it's not too bad, but it's something that we need to take into account. If we know how to do that, we know how to explore the set of objects. And so here I'm just giving a small example in our case, like the smallest one we could build. We have 12 samples and 3-node compacted double graph. We want to test at some level alpha. And we want to test all the possible connected subgraphs. Here on each of the nodes, I'm giving the mock presence pattern. So remember, each of these nodes is a sequence. And here, this is the presence over the 12 samples of the sequence. Okay, they're not the same. And so assume we start from node 3 here. We test if it's associated with. Sorry, we don't test it. We just compute its minimal, the minimal p-value for this one, and we obtain p star. It's 0.06. Okay, since k for the moment is 1, alpha over k is 0.15. It's our threshold, and so v3 is testable. We add it to our active set. We do the same thing with 2. The minimal p-value is 0.004, and so both here. Sorry, the V2 is testable at level alpha divided by 2, which is 0.75. Sorry, both of them are testable at this level, so we're fine. And then if I want to add a third one, k over 3 now is 0.05. And you see that V3 here, which was originally testable, it was testable at level 0.15, but it's not testable at level 0.05 anymore. So I have to remove it from my set. K is still 3, but my threshold is lower. V1, its minimal p-value is 0.004, so it's still testable at this level. So now my set is V1, V2. So I've done all the individual nodes, and now I will keep exploring the connected subgraphs. Normally, I would start from V3 and see all the subgraphs that contain V3, but I removed V3 from my set of testable subgraphs. So I know that any subgraph that contains V3 is not testable either. So I don't have to explore all these parts of my tree. So I start from V2. From V2, all I can do is add V1, and so this ends the enumeration. I have V1, V2, and the pair V1, V2. Okay. So it's probably hard to follow unless you already know the procedure. in detail, but the idea that I want to show is that by doing that, we managed to prune. We don't have to explore explicitly all the possible subgraphs. So there is something I put under the carpet for the moment, is that what we really want to test is the association between a trait and a variant, which are both measured by a vector of size n, where n is the number of samples. And I've talked about connected subgraphs for the moment, but very often the compacted double graph will look like this here. So that's the same idea as the picture I was showing. There was lots of small bubbles because maybe all the first 3 samples here have the gene, but some of them will have a different version. So this united here will only be in the first 2, and the third one will have a different version here. Okay, so you'll have lots of small bubbles like this. And if we start from either end of this or any intermediate node, and we start adding nodes, very quickly, all the connected subgraphs will have the same pattern, which will be 1, 1, 1, 0, 0, because we're taking these junctions, right? So I start from here. This junction by adding these 2 is any of these 2 is the same. This junction by adding this one is the same, etc., etc. Okay, so will be the first 3. So this means that I'm going to, by doing this exploration of the connected subgraphs, I'm going to redo the same tests a lot of time in lots of cases. So the correct objects that we want to enumerate are not the connected subgraphs, they are the closed connected subgraphs, which are the subgraphs whose presence pattern doesn't change if I add any of the neighbors. Okay, and this was probably the hardest part actually of this contribution of this work, was to enumerate efficiently this set of objects. The principle is the same. We also used reduction, but the inversion of the reduction is much more involved. So the idea, you could generalize what we did for connected subgraphs, and instead of saying my reduction, remember the reduction for connected subgraphs was just, I removed the node, it's the same, I just highlighted in red the part that will be changing. So it was, I removed the node with the largest index such that I don't disconnect the graph, okay? If I want to do the same thing, but not get a smaller subgraph, connected subgraph, but a smaller close connected subgraph, I need to remove nodes until I change the presence pattern. Otherwise, the closure is still the same. And then take the closure because I want to have a close connected subgraph, right? So the reduction is not that complicated. I will just, instead of removing one, I will keep trying until I change the presence pattern. But the inversion is going to be very costly. Because, well, I won't go into detail, but I need to maintain a set of articulation points that will change a lot of times. The idea that we used is kind of desperate because, well, I will explain why, but it was to say we need to forgo connectivity. If we want to maintain connectivity in the reduction, the inversion will be way too complicated. So we have to forgo it. And so we remove nodes that change, a set of nodes that change the pattern, and maybe it will disconnect the graph, but we will retain as a parent only one of these connected components, and we'll define it properly. Okay, because we do that, the inversion does not require to check anything about connectivity, so it's much faster. But it comes with a cost. It's that it's harder to define the possible set of children. You can check if a particular subgraph is a child, that's easy, but reconnecting all the possible children is a bit more involved. Okay, I don't go into details about that, but that was the hard part of the inversion. And so this is, when we put everything together, we have a full procedure for testing all the close connected subgraphs. And if we apply it to the same data we used for dbGWAS, which was amikacin resistance GWAS and Pseudomonas aeruginosa, this is the first hit that we obtain, which is a plasmid with resistance genes. If we apply dbGWAS at the same level on this data, then we only find small pieces of the plasmids, which are very hard to reconnect. So this is compared to dbGWAS. If we use more, if we want to do the same test, but we use more naive exploration methods, than what we proposed. Then instead of running in 3 hours like our methods, it will never finish in a reasonable time, because in 24 hours we had only explored 2% of what we managed to do with our method. So just to summarize, we've used close connected graphs of the compacted double graph to define genetic variants. We think it's providing a better scale than unitigs for exploring genetic variants because it's less prone to dilution effects. It leads to very bad computational and multiple testing issues, which we're addressing using the Taran procedure combined with a reverse search strategy. And thank you for your attention.
Thank you. Thanks a lot, Laurent. So we have time for a bit of questions. Yes, you have a few minutes. Unless there is a question in the audience, I may start with a small one. A question about application. So if I understand correctly, today Caldera is able to analyze data based on 2 conditions.
Yes.
Do you have an idea of how it can be extended to apply on QTL or to time series or something more continuous?
Not directly, no. Unless either you can discretize it or there is no obvious solution, because the whole testability thing depends on having discrete tests. So for DBGWAS, for example, we started with binary phenotypes, and everybody was asking, can we do it with continuous phenotypes? And it was obvious that, I mean, it was not a bad question, but it was very easy to generalize to continuous phenotypes. But here we really rely on the fact that we have a discrete tests. There may be solutions, but they are not obvious.
Okay. And I continue if there is no other. Okay, so this pruning procedure you propose seems to be very efficient in practice. And do you have some theoretical estimation of the theoretical gain offered by this procedure?
Theoretical, you mean in terms of power or in terms of speed?
In terms of the quantity of space you do not have to explore, thanks to this Taran procedure.
It's a good it's a good. We could have done it. So I don't have a theoretical, because we didn't pursue that too much, but we would require to know the exact number of close connected subgraphs that are present in a compacted double graph, and we don't even know that. Maybe there are ways to estimate it or bound it, but we didn't fight on that. No. Mm-hmm. So it's just empirical. We just see that we managed to run the method on the data set, and if we don't do it as efficiently, it's hopeless. But we don't know how hopeless it is.
Mm-hmm. So this means that you cannot anticipate how well it will work depending on the complexity of the data sets?
So actually, we know that on current relatively small bacterial data sets, it's already not enough, and we need to prune more aggressively. We are using heuristics. Like we stop exploring when we have too many nodes. At some point, we say, okay, this is too big. It's not interesting for us anymore. Anyway, whatever this is, so we don't explore. And also, we stop the exploration at some point. We have a heuristic for that because otherwise it's too long.
Okay.
I think Alex was raising his hand and had a question.
Yes, I would have a question, but also in the chat there is a couple of questions raised already. Yeah. Maybe they were before me. Maybe someone can read them from the chat. Just.
So maybe I can read the one from PO, the first one. What do you consider are the limits on number of samples genome size?
I don't. Well, all I can say is we had a few hundreds of genomes. Genome size is not necessarily the most the best thing to look at, because if you're familiar with k-mer GWAS, what's really important is the number of k-mers. So if you have lots of MTB genomes, for example, they are almost all identical, so it doesn't make a big difference. If you have a small number of E. coli, then it becomes very bad very quickly. But yeah, to get some intuition for the GWAS that we did on Pseudomonas, we had a few hundreds of them, and we already had to use the heuristics to stop the exploration. So probably you can still use the tool on bigger with more k-mers, bigger genomes. I don't know, but you will have to stop the exploration earlier. It's probably still better than just looking at the unitics, which is an extreme case of stopping the exploration at just the node level. It's just that you won't be able to do the full exploration. My intuition is that it's still slow. It's not scalable, not very well scalable, but it can still be interesting to look at to find things that you would not find with unitics. Another thing with the number of samples is that the more samples you have, the smaller the minimal p-value will be, so you'll have less pruning too. That's something to consider.
Okay, thank you. And there is a question technical from Codor about the typical size of k-mers.
Yes. So we didn't, for Caldera, we didn't explore it too much. I think we use 41, which was also what we use for DBGWAS. For DBGWAS, Magali spent some time trying different values. I'm still sharing my... You still see the slides?
Yeah, we do.
Okay, so yeah, she did some tests, and so the quick answer is that there is a wide range of cases for which the results are robust. Okay, we use 31 or 41 depending on the times, but you get very similar results for that. If you use a K which is too small, then the k-mers are not specific enough, so it's very hard to make sense of it from the debugging graph. If they are too large, and we'll include haplotypes with several variants in them. So it will be harder to interpret, to lose some resolution by doing that. But yeah.
Okay,
so I would have a question. Yes. So I was wondering at the beginning, you were saying that the reviewers were saying, well, or like with the early approach, okay, you can run it on Earth, but what is the meaning of a k-mer? Yeah, and here you now test against connector subgraphs, and I was wondering whether you don't care about the shape of the subgraphs, because otherwise you may end up with subgraphs that are really hard to interpret again, or do we have a misunderstanding maybe?
No, you're right. Ideally, we would like to restrict ourselves to the small strings that we're seeing. That's our motivation and what we want to find. So yeah, we could use some heuristic that says we stop exploring if some statistic of the shape of the graph is we don't like it anymore, if it's too connected, for example. That would actually be a good idea. We hoped that this would be taken care of by the threshold. Like if you have something that doesn't make sense anymore, then it's going to include too many samples and not be related to the phenotype anymore. So it's not going to be not going to pass the threshold anymore.
Okay, but at the moment you consider everything. So this is
yes, but you're right. That could be an improvement if we had a way to say, okay, this we don't like the shape of this subgraph, we don't consider it. That could be a way to prune more aggressively, maybe.
Okay, thank you.
Thank you.
Are there any other questions? There was a remark from Benjamin Lina on the chat about the size of K related to the distance of variants. and the genome?
It's a probably a good point, but I think the issue is that you would need to know the. I don't know if the self-resistance is always known. If you want to define mutations in the genome, you need to have a reference probably, and this is what we wanted to avoid in the first place. But I guess in practice, maybe in lots of species that we look at, this is something people have a sense of and we could use. Also, this is probably good for SNPs, but I don't know if it's the best possible scale for finding genes, for example, mobile genetic elements or plasmids. Thanks.
Okay, so if there is no other question, I would like to thank you again warmly, Laurent. So thank you.
Thank you. Thanks, everyone.
And so this was recorded, so we will put the talk on YouTube. And thanks again, and see you soon all.
Thanks, everyone. Bye.
Ciao, bye-bye.
Thank you. Bye-bye. Thanks a lot.
Thank you. Bye.